Okay, so to do a quick review of what we've done so far, when we're looking at dominance and recessive, you have two choices. It's either going to be one or the other. Now, when we're talking about dominance or recessive, we're talking about two alleles, and they're both going to be present. And the question is which one is going to show up, and that's going to be determined by which one is dominant over the other. One covers up the other gene product. So when you're looking at the phenotype, if you have a heterozygous individual, one allele is going to be dominant over the other, and so you have one or the other. That's dominance and recessive. Dominant, recessive. When you're dealing with incomplete dominance, you have neither. There's a blend between the two. And we're going to look at a couple examples of incomplete dominance and codominance in just a minute. Now, codominance is both of them show up. You don't get a blend, you have both of them showing up. Now, here's an example of incomplete dominance. Now, I want to emphasize that the notation looks different. And different books use different notations. Some of them will use the same, a big R for red and big W for white and P for pink. So they'll use three different letters. This is the most common and the most accepted way of notating incomplete dominance, and that's the idea that you have here the W, all right. So C is the color of the flower, and W is white, where red is going to be the R. And then when you have an R and a W, you end up with a pink. Now this is a blending, so this is an incomplete dominance. And you'll notice that this is a heterozygous, one CR color red and one color white gives you a pink color, where two white colors, two W's, give you the white color, and two reds, if they show the genotype for the red, would say C big R, C big R. When you take the eggs and sperm that this pink flower can make, you'll see that it can make a CR and a CW because that's what it has. Both the sperm, if we're doing two plants from the same generation, we'll take the pollen from one plant and the eggs from the other and mix them together. We'll see that we'll get CR and CW. This would be, again, from the pink plant. And we have heterozygous, so we expect not the 3-to-1 that we would have in dominant and recessive, the traditional Mendelian genetics. What we would then see is not that, but we'll see a red, big R, C big R, big R. That'll be our red. We'll see 50% is going to be big R, big W. All right, so that'll be pink. And then we'll have 25% white, C big W, C big W. So what happens in incomplete dominance is that the genotype and phenotypic ratios are the same. You get a 1-to-2-to-1 ratio. Now, if you go back and look in your notes and look at dominant and recessive, what you'll see is that you'll have, for phenotype, in the traditional Mendelian genetics, what you'll see is in the phenotype, you'll get three of them being purple, let's say, and only one being white. But in the genotypic ratio, you would get one of the purples being homozygous dominant and two of them being heterozygous and one of them being recessive. Now, because the heterozygotes don't look like the homozygotes in this incomplete dominance, what you end up seeing is that the phenotypic ratio and genotypic ratio are the same. So you get 1-to-2-to-1. 1 big R to 1 big R W to 1 WW. All right, so that's incomplete dominance. Again, it's blending. It's the idea here is that we're dealing with a blending of the colors. All right, we're not dealing with one or the other. We're dealing with a color combination that comes up with, it gives you a third color. All right. Now, the classic example of codominance is a ABO blood group. The ABO blood group is really based on the idea that you have these antigens on the blood cell that are proteins, the round ones being representing the A antigen. It's a protein on the cell surface like we've studied already earlier in the year. And antigen B is a different protein, a different shape. Remember, we talked about molecules having shapes and that these shapes are very important. And you see that antigens, when you have both antigens, A and B, they both show up. There's not a mixing of the two. There's a distinct round shape and a distinct triangular shape. And then having neither A nor B means that you have, of course, nothing on the cell surface. Now, when you put blood into someone's transfusion, when you do a transfusion, which, by the way, was first researched by an African American surgeon researcher, Charles Drew, what they looked at was these antibodies that are in your blood that respond to foreign material. Now, if you have a blood type A, you're not going to have the antibody to A if your body is normally working. You're going to have antibodies to B in your blood. If you have B blood group, you're going to have the antibodies to A. If you have AB blood type, you're not going to have either antibody in your blood. However, if you have neither A nor B on your blood cells, in the membranes of your blood cells, you're going to have both A and B antibodies, which means that if you're O blood type, you cannot take any of the other three blood types. If you're A, you can only take A or O, because O, as you see, has no antigen, so it will not react with the antibody. The opposite antigens, or the antigens that are made against, the antibodies that are made against the particular antigen, are what trigger the immune response, the killing of any foreign object. Killing is, the destruction of any foreign object in your body. And so we see that the bottom line is that A can only give to A or AB, but can only take from A or O. All right. So this is the ABO blood group and how it works. That's not the most important thing here as far as the genetics component of the proposition. It's that we have this example that when you have the allele, here we have the allele for A, you'll notice it's the same notation that we looked at just a moment ago. If you have the allele for A, two copies of the same allele, then you have blood type A. You're only producing this A antigen on your cell. If you inherit an A from mom and a B from dad, then you end up with AB blood group. You have AB antigen, both A and B on your cell surface. Your body, therefore, will not produce any antibody to it. So it can accept blood from either A, B, or O. No, there's no, so the AB blood group is known as the universal acceptor. So if you have the little I is broken. So it's not really a codominant situation. This is, if you want to consider it a recessive trait, it's the O blood group is that gene not working at all, not producing any protein, where A and B are producing. If you get A, you produce A. If you get B, you're producing B. If you have little I, you're not producing anything. So it's a little complicated, but it's kind of classic because it really shows you exactly how codominance works. In other words, if you have the allele, it shows up. The exception, of course, is the broken allele I. So you have, and I use the word broken loosely, just to try to give you an image to think about. It's just an allele that does not produce a protein on the surface of the cell. So you see, if you have double A, you have A. You can get A if you have A and little I. So if you have two little i's, of course, you have O blood group. And what you get is this kind of, this type of Punnett square where you have three possible alleles versus three possible alleles. It's a really good example of codominance. I just wanted to give you one really good look at codominance and at the idea of incomplete dominance. And flowers are the perfect example because the color patterns are so clear. So you can see here that in this flower you have the red sections and the white sections. They both. So if the gene, this flower theoretically has the gene for red petals and the gene for white petals, and both of them show up. Red is red and white is white. There's no mixing of the colors. It's just if the gene is present, the color is going to show up, just like ABO blood group. So we call this codominance. Here's another one as another example of codominance where it's red, and where it's white. There's no mixing of the colors or blending. Now here you see a white flower which has the double white gene. Here's a red flower with a double red gene. But here's the flowers with pink. So the two colors are blending together. Here is an example of incomplete dominance. So make sure you know those three ideas, or four really. You have your dominant and recessive. That's a classic kind of Mendelian example we've been looking at for about a week and a half. That's where you have purple flower, white flower. The heterozygous is purple because purple is dominant over white. Okay? So that's your class. That's number one. That's the one or the other. You have incomplete dominance where you have blending, which is what Darwin was trying. Darwin's work initially brought the idea of blending into question because before Darwin, everyone thought that all inheritance was blending. We know today that that's not true. There's several different ways of doing it. Darwin described the one or the other, the dominant and or recessive, okay? So when you're dealing with codominance where both of them are showing up, now you're where in the incomplete dominance you have neither, right? They're both mixing and you're coming up with a third option. In codominance, they're both showing up. So it's and. So you have an or, a neither, and in this case an and. You have red and white showing up. You have red and white showing up in the flower. So those are three major inheritance patterns that we've discussed thus far. That takes us through sections 6-1 and 6-2. Make sure you know how to do the Punnett squares for those. Do the workbook problems, and you'll be ready for the quiz and the exam. I want to make sure I put out tomorrow a discussion, a description, an explanation of the case studies that we've covered so far, and that should take us up through next week, Monday. Now make sure you're ready, getting your work done, and do your study island for physical science because you're facing that park exam the second week, or I think the first week of March. And of course, next week you'll have your math and English park exams. All right, good luck to you.